my name is Corey Padilla, and I'm here with Dovetail Genomics. And today, we would like to demonstrate how you can capture the epigenetic and 3D genome architecture signals that are found in Hi-C data while still preserving all the capabilities and applications that you would normally perform with whole genome sequencing data. So the idea here is that from a single Omni-C library prep, you'll be able to do large structural variation detection, you'll be able to do SNP and InDel genotyping, you'll be able to perform genome assembly and haplotype phasing, and again, you're still going to be capturing that 3D genome architecture that's found in Hi-C data. So the reason that Omni-C can support so many whole genome sequencing applications is that we've been able to mimic the genomic coverage found in shotgun data, which is the dashed line in all the figures that we're showing here. And all the rows are different Hi-C approaches. So what we're showing here in the top left figure is that blue line, which is the Omni-C genomic coverage, almost matches shotgun-like coverage. And then we also don't enrich the sequence space in restriction enzyme sites, which is commonly found in other Hi-C approaches, meaning that the Omni-C dataset is almost shotgun-like when compared to other Hi-C approaches. Now, this improved genome coverage allows us to call SNPs and genotype at a fidelity that's almost equal to that of shotgun data. Now, with that improved SNP calling combined with the Hi-C data, we're able to phase chromosomes end to end, which is something that shotgun data on its own can't do, but we have a much lower switch error rate when compared to other Hi-C approaches to phasing. Also found in Omni-C data are large structural variants. So here what we're showing are an inversion, a deletion, and a translocation event that are in the breast cancer cell line HCC 1187, where we're pointing out the breakpoint with the arrows, and then we zoom in on those breakpoints to be able to see the genes that are associated with those breaks. So just to summarize, in a single library prep, you'll be able to capture the epigenetic and 3D genome architecture signal while still preserving all of the applications and capabilities of shotgun data that you would normally use for whole genome sequencing studies. So if you're interested in any of the applications shown here, please reach out to us and we'd be more than happy to answer any questions. Thank you.